Do you never listen to me? I literally just said it. All right. So today we're talking about it's Unit 2. That's what all of this is Unit 2.
DeMarco.
What did you expect it to do?
Oh my god.
Unit 2 review. All right. And I'm starting over at the very, 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 very beginning of Unit 2, which starts off with prokaryotes versus eukaryotes and organelles. And organelles and cell membranes and all that stuff. Okay? So, if I were to look at the very beginning of all cells, it starts with compartmentalization and stuff like this. And so, there's 4 things that all cells have. Nope. Okay, all cells have 4 things. All cells are going to have, number 1, a cell membrane. No matter what, whether it's a eukaryote, whether it's a prokaryote, whether it's a plant cell, whether it's an animal cell, every single one of them will have that. Number 2, I don't know, want to add something. They're all going to have ribosomes. They're all going to have ribosomes. Okay? Number 3, they're all going to have cytoplasm. Cytoplasm/cytosol. And number 4, they're all going to have genetic material. Okay? Keep in mind, I'm recording this, and I'm going to just post this directly online. So if you don't want to have to write this right now and you want to do it later on in your own time, you can. But go with how you feel. Go with what makes your heart happy.
If I don't do it, I'll forget about it.
You're right there, my little ball of string.
Wait, so you're like, can I...
Humor me, DeMarcus. What question do you have? You're about to knock off that scale, by the way. Thank you. That's the question you have? Of everything you can have?
This is going to be recorded, right?
DeMarcus just walked into a chair, and now he's running to the restroom. The phone is ringing.
Can Simon come to the classroom?
I don't care.
Yeah, he can come.
You're much friendlier than me. Okay.
No, he asked,
He asked.
if he can.
Next thing.
That sounds like a Simon.
Who called?
Figure out.
Oh.
So that Simon can be.
Did she say her key term?
No.
Oh my gatos.
No.
Dude, that's how I got to saying that. Because of her.
That's Fig's call.
Yeah, me too. Yeah.
Oh.
Okay. Prokaryotes. So prokaryotes are extremely small. So they're extremely small. They do not have membrane-bound organelle. Okay. There's no nucleus. So since there's no nucleus, genetic material like DNA or RNA, any of that stuff is in the cytoplasm. So I'm just going to put GM in cytoplasm. And finally, these are the oldest, most basic things. These are the most archaic of them. Not most. Okay, so prokaryotes, most commonly prokaryotes are bacteria. They're single-celled bacteria. That's what we're going to get to when we get to the organelle. It's one prokaryote ate the mitochondria or ate the chloroplast and gave rise to those organelle. So the mitochondria and the chloroplast at one point in time were their own. Genetic material. All right. Ready for eukaryotes? Okay, so eukaryotes are much larger. They have membrane-bound organelle. Right there. Thank you. And they have a nucleus. Woo.
Why'd you cross this off?
Because I talk about that right here. I didn't want to have it to be redundant. All right, so let's start talking about some of these organelle. All right. So these organelles are small compartments with jobs. So there are these little structures within inside of a cell that it says, hey, you do this. You're not going to freeload off me. Okay. The first one is the nucleus. Do you know what's housed at the nucleus? Yeah. Good, thanks. I'm glad you know that. What is it? Okay. Houses genetic material or DNA. Okay.
Oh.
So houses genetic material like DNA or RNA. Someone's getting down with some Kahoot.
Yeah.
I hear some Kahoot.
Like major anxiety. Like I can feel my heart racing.
For a while there was a ring of kids on campus who anytime they had a massive group chat, and anytime one of them played Kahoot in class, they would send out the join code to all of their friends in their other classes. So instead of like the 32 kids in one class, there'd be like 80 people signed into the Kahoot. And none of the teachers knew why. And people would just randomly just rack on symbols. All right. Ribosomes. Don't talk about me when I'm right here. Ribosomes. All right. The thing about ribosomes, they're special because they're not membrane bound.
45.
They're made of ribosomal RNA and proteins. But ribosomes are not made of membranes. Like a lot of these other organelles are, ribosomes aren't. Any clue that a ribosome is not made of a membrane-bound organelle? Look at the things that all cells have. The four things we said. What are the four things we said all cells have? Cell membrane, ribosomes, cytoplasm, and genetic material. Okay, so all cells have ribosomes. What was requirement, or in prokaryotes, what was thing number 2 we said? Does not have membrane-bound organelle, but does a prokaryote have ribosomes? Heck yes it does, because all cells have it. And the prokaryote's a cell. All right, so it has this ribosome, but this ribosome is not membrane-bound. This ribosome is actually made of rRNA. Okay, and so our next one we're going to talk about is the rough ER. Whoa. Go away. I don't want apps or people. I don't want people in my life. Go away. There we go. Rough ER.
What does ER stand for?
Endoplasmic reticulum. Sorry, I'll write it out. Okay, I'm going to put ER. So it is where proteins are made for outside of the cell. Proteins that are going to be shipped out of the cell are assembled at the rough ER. Yes, ma'am. Absolutely. Because they're the simple, most basic ones. Prokaryotes are. They're the most prehistoric, the most ancient things. And so they don't require that much because at the beginning of life, so when life kind of started about 2.5 billion years ago, there wasn't much. And so those are the most ancient ones. Okay. So if we're going to export a protein out of the cell, it has to be made at the rough endoplasmic reticulum. All right. I'm going to put a little asterisk here. All right. If protein stays in, it's assembled at a free-floating ribosome. It's not assembled on a ribosome attached to the ER. If protein stays in cell, assembled on free-floating ribosome. Because we can have ribosomes just floating around in the cytoplasm. If this protein's not going anywhere, I don't need it to be bound to the endoplasmic reticulum. All right, I can do it right there. We've got another type of ER. Remember what that other one is? Smooth. Smooth ER. All right. This is going to be where it detoxifies and synthesizes lipids. I'm going to put synthesize because you need to be familiar with that word. Synthesizes or makes lipids. This is where fats are made. Therefore, my smooth ER is pretty busy. Hurt. Okay.
You're lying here, so.
You guys doing all right? Hang in there, Ninos. Hang in there.
How are you doing?
I'm thriving.
Good. You deserve it.
All right. Luis Amaral. Fall rosters. What the heck? I don't need y'all to see the fall rosters. Then we get to our next one, the Golgi. Golgi body, Golgi complex, Golgi apparatus, just Golgi. I'm gonna put Golgi complex, Golgi complex. Okay, but as long as you're familiar with the word Golgi, you will see it called several different things. As long as you see Golgi, Golgi will be in there the same thing. So this is gonna be modify and package proteins. Anyone remember what they package it into? It's a special container made of a phospholipid bilayer. Close. Same starting letter. They're commonly just vesicle. Yes. I know. I do it all the time too, where I say vesicle when I mean vacuole and I say vacuole when I mean vesicle. But I knew you were on the right track, the fact that you had the V. Package into vesicle. All right, so here's its little phospholipid bilayer mailing package container, so that way it can be exported. And then we get into, we'll save those ones. We're gonna lysosome. Okay, still membrane bound, contains acidic. If something is considered polyamin, I don't want your floral arrangements right now. Okay. If something's acidic, it means it has a really high amount of hydrogen ions. Okay, that's what makes something acidic. If it has the more hydrogens it has, the more acidic it is. So it contains acidic hydrolytic enzymes. What's hydro mean? Water. What does lytic or lysis mean? To cut or to break. So it contains enzymes that are going to use water to cut or break something down. So it's very acidic, but it's going to break these things down. Breaks down waste.
Something from Universal Studios.
I don't know what is there.
Me neither.
You don't need to give me anything. You just enjoy you. You spend your money on you.
Nope.
Yep.
I want.
I don't need anything.
Yeah,
Vacuole. This big thing's going to store nutrients in water. There's two big organelles we haven't talked about yet, and Alex mentioned them earlier. Okay, they're two little special ones. Okay, well, they're not little, they're very freaking important. Number one is the mitochondria. Okay, and this mitochondria, it's going to be where ATP is made. Okay. And if we looked at the structure of it, like I said, it kind of looks like this bean. But it's got this right here. So we've got a smooth outer membrane, highly folded inner membrane. That's supposed to say membrane. Good luck. Can you see any benefit to having a highly folded inner membrane? We kind of talked about this when we were first talking about it. There's a benefit for it having a highly folded inner membrane. It what? It's got a higher surface area. So higher surface area equals more ATP. Simidenji. Hey, bud. How you doing? All right. Then we've got, yes, ma'am. So hold on. What? Sorry. What does it combine food particles with? It's going to take glucose and it's going to break glucose down to make ATP, and one of the byproducts is going to be CO2. It's actually what our next unit is all about, energetics. Yes, ma'am. High surface area equals more ATP. High H's, so high hydrogen ion concentrations.
What's the 15 for?
Huh?
What's the 15 for?
What's the what for?
Apostrophe S.
Oh.
High hydrogens. So something is acidic based on the number of hydrogens it has. The more hydrogens it has, the more acidic it is.
I was sitting down.
I'm so confused. What are you talking about?
This 15 right
The
here.
apostrophe S
That's an
looks
apostrophe
like
S.
That looks like a
looks like
15.
You look like you're 15.
I'm 16, close enough.
I am a little kid.
Okay.
You consider a little kid drugs?
Yeah.
So-
Marco, you took a while in that bathroom, huh?
Yep, I had diarrhea.
Did you put down a landing strip so you didn't get Poseidon's kiss?
You're recording this, aren't you?
Yep. All right.
Oh, I did not have diarrhea.
This is
All right.
still the small compartment for jobs, right?
Huh?
It's still underneath this bracket.
Yep.
Organelle.
This is our last organelle. So the chloroplast, it's also this double membrane structure, all right, but the inner one is not highly folded. This one's not making ATP. This one's making glucose. So I can turn around and make ATP from the mitochondria. Every cell that has a chloroplast also has a mitochondria, but not every cell that has a mitochondria has a chloroplast. So for instance, when we associate chloroplast, what do we usually think about?
Plants.
Plant cells. Plant cells have a chloroplast. Plant cells have a mitochondria. All right? Animal cells do not have a chloroplast, but they have a mitochondria. The reason why, if I need to make energy, I can just go to Safeway and I can get some food, eat it, and make energy from that glucose. A plant cell cannot. It's stuck. All right? The thing about both of these, and what some of you are asked, or what Alex asked about earlier, and you may have been like, huh? What are they even talking about? These two things right here, this, yes. These were once their own prokaryotic cells. It's called the endosymbiotic theory.
Oh my.
It means at one point in time, they were a prokaryote. We had another prokaryote that ate it. All right? For those of you in sixth period, you don't know what it is yet, but it underwent that process of endocytosis. So it ate another organelle because it needed nutrients. It brought them in through active transport through endocytosis. It ate it. Normally it would digest it, but this time it says, hey, I'm not going to digest you, but you're going to have a job. So for instance, you might make food for me, or you might make energy for me. All right? And so they started living together symbiotically. All right? Thus the term endosymbiotic theory.
Oh my gosh, man.
Okay? So one engulfed the other. So another prokaryote engulfed the mitochondria, or it engulfed the chloroplast, and they started living together. All right, symbiotically. That means they get along. And so our evidence for this is the mitochondria. I'm just going to put the mito and the chloroplast. Both have cell membrane. So they both have the cell membrane. They both have ribosomes. They both have genetic material. And they both have cytoplasm. You remember the four things that all cells must have?
Ribosome.
You want to try that one again? Ribosomes, cytoplasm, genetic material,
And cell membrane.
and a cell membrane. The mitochondria has all four of these. The chloroplast has all four of these. That's our evidence that these things at one point in time were their own cells. They existed at one point in time on their own. And then they were engulfed by another thing and they started living together. That's the endosymbiotic theory. So, if you break down those words, what does endo mean? Inside. Symbiosis or symbiotic means getting along. So they live inside of one of the other one and they get along with each other. They both benefit.
Oh, like a pregnancy.
That's a parasite.
Okay.
Okay?
That's funny.
All right. Any questions about any of these organelles? Tomorrow we're going to talk about in Bulldog Block. We're going to do membranes, transport, all that stuff, structure. Okay. Like I said, I recorded this. I'll post these notes on Classroom if you need them, along with the video.
You
Like
should
and
make a YouTube channel. I don't know why you don't.
It's like was not
Like
recording.
a traveling.
Recorder is recording. Click here to.